Hey everyone, welcome to Biology Lab. I hope you're ready for a very tastic experiment today. You're going to be extracting DNA from strawberries, and at the end of this experiment, you're actually going to be able to see DNA with your naked eye, which is pretty incredible. So we're going to be using strawberries today because they work really well for this experiment. They have about 8 chromosomes, and it's a very nice number to work with. And today we're going to be breaking open those cells and allowing the DNA to spill out, which is how you're going to be able to see it. Before we do anything, let's go ahead and pose our question for the experiment, which is, what does DNA look like? Now that we're ready to start the experiment, let's gather our materials. I have all my materials in front of me here. If you can take a second, pause the video, and go ahead and grab your own, and then we'll get ready to start. So now that we have everything that we need, we're going to go ahead and create the DNA buffer solution first. Get that out of the way. So you're going to need about 1/2 cup of deionized water to start with, and a tiny squirt of dishwashing liquid. It can be any brand, anything, as long as it's clear, so that way you're actually able to see what's going on in the experiment. And we really just want a little squirt, not too much, and a pinch of salt. And then you want to go ahead and mix it together. And this buffer solution essentially is going to help break apart the membranes so that the DNA can be exposed. So now that that's mixed, I'm going to go ahead and move that to the side, and I'm going to move these materials over. And now we can go ahead and focus on our strawberries. So we're going to go ahead and take about 5 to 6 strawberries, should do it. We're going to need 1 Ziploc bag. And what you're going to do is you're going to take the strawberries, put them in the bag, and essentially mash them as much as possible. You can do it with your hands, just as I'm doing. Mash them up as much as possible so that the juices are coming out. So this step is important because we want to mash up the strawberries so that the plant cell wall is broken down and the cellular membrane is exposed. This is really important because if you remember, plant cells have an extra cell wall that keeps them safe from external environmental factors. And so what we want to do is break that first barrier down so that way we can get a step closer to the DNA. So I'm going to go ahead and move my box of strawberries. I now have a good amount of mashed up strawberry in my Ziploc bag. And what we want to do now is we want to take our buffer solution and add it into the Ziploc bag. And what this is going to do is the soap in here is actually going to break apart the membrane. So if you remember, the membrane is made of phospholipids. So is the soap. So they want to interact and intermingle. So when they do that, they're actually taking the lipids and proteins out of the cellular membrane. That is going to cause big gaps in your cellular membrane. And that's how essentially the DNA is going to be exposed. The cool thing is that the cell membrane and the nuclear membrane are essentially made of the same thing. So both of those barriers are going to be broken down once we mix this up. So we're just going to go ahead and mix it up. You should see a few bubbles forming from the soap. That's perfectly fine. All right, now we're ready for the next step. So what we want to do is essentially filter this out. So we're going to take a coffee filter here and a nice clean cup. place your coffee filter on your cup, not all the way down, so that the strawberries have enough space to get filtered through. And we're just gonna pour our solution in, oops. And let it soak through. If you have anyone at home that can lend an extra hand, you can have them hold the coffee filter for you while you pour the solution. If it's just you, like me, then we'll go ahead and take it slow and wait till this filters out and then add in more solution. And in the end, you really don't need a lot. Less than 1/4 cup is perfectly fine. Alrighty, so your solution should look somewhat clear. If it's not fully clear, that's perfectly fine, but most of it should be strained out. If you've got bubbles in it, that's fine as well because your buffer solution is mixed into it. So I'm gonna put my extra strawberry extract to the side, and what we're gonna do now is add just a few drops of pineapple juice. And pineapple juice has a lot of enzymes in it. And what these enzymes are doing right now is they're taking the proteins inside the DNA and taking them out. They act essentially as scissors. And so they're cutting out all the proteins, which is unwinding and unfolding all the DNA. So once it's unfolded, it's longer, and that's how we'll be able to see it. So now that that's done, we're going to add some isopropyl alcohol. This should be chilled as well, and so temperature here really matters. The colder it is, the better it acts as a separating agent, and so the DNA is able to clump together. So we'll pour about up until halfway. You want to slowly pour it in and tilt your cup just slightly, so that way it forms a layer on top. Do not mix the mixture. You want to keep it separated. If you can see up here, there is a white filmy substance immediately forming in your alcohol layer. That is your DNA. If you give it a few minutes, it'll start to clump together. The salt in here helps it clump together. It neutralizes the DNA. In a couple minutes, you'll be able to take a stirring rod or a straw or even a spoon and pull this substance out. So your DNA, you'll see it's very quickly clumping up together. And what I'm gonna try to do now is take my straw and just lift it. Ooh, it's gooey. And when you do this, you'll be able to see all of the DNA up top. You can see it's white, it's slimy, it's gooey. You could touch it with your hand if you want to. So there you have it. You have your very own DNA that we didn't even need a microscope for. You can try this at home with various items. You can use split green peas, you can use spinach, you can even use your own saliva. Go ahead and spit into a cup and you can take a look at your own DNA.